Sir, second session is to be chaired by Dr. Hajime Kojima.
Okay, Dr. Kojima will be chairing.
Hello.
Okay. Dr.
Yeah.
Kojima,
Yeah.
go ahead, please.
Yeah.
And
Hello.
over to you.
Can you hear me?
Hi, Kojima.
Yeah,
Nice to
yeah.
see you.
Hello.
Nice
Yeah.
to see you.
Hi. Good afternoon. Nice to meet you. Yeah.
Right.
Yeah, I'm very happy to the hot discussion in the OECD test guideline. Yeah. Thank you so much. Yeah, it's a great pleasure to give you an opportunity to Japanese colleagues in the Society of Alternative Animal Experiment in India. So in the previous session, the JT colleagues to talk about their own human epithelium and cornea model, its test method. And based on this presentation, I am happy to discuss it to the Indian colleagues at OECD and also to regulatory acceptance. Okay, thank you. And so now, Kao's colleagues will talk about the new in vitro skin sensitization assay. So Dr. Mizumachi and Dr. Suzuki, they is also to talk about the epidermal sensitization assays, epicensor in vitro skin sensitization assay based on the gene expression reconstruct human epidermis. So they research scientists of skin sensitization of Kao Corporation, and also they develop a developed a non-animal test method like the H-graph. And also they are developing a novel method and strategy for prediction of skin sensitization potential of chemical. I think that this year to develop by the DAS in OECD. DAS means so defined approach of skin sensitization in this years. So this guideline, including the H-graph and so to DPRA and also keratosense, is a combination of the test method. So based on these test methods, so Kao's colleagues to develop the new type of skin sensitization assays. So Dr. Mizumachi and Suzuki will talk about the new test method from now. Okay. So Suzuki-san, can you hear me?
Okay, thank you.
Okay.
Thank you.
So Mizumachi-san is also first speaker is Suzuki-san.
Yes.
Yeah. Okay. Please go ahead.
Okay. Thank you for introducing us. Oh. Can I cannot share my slide.
Mr. Suzuki, your sharing function is activated or not?
Not.
Yeah,
Not.
okay. I have activated you now. Please check now. Please try once.
Okay, thank you. Can you see my slide?
Yeah, that's visible now.
Okay, thank you. So I'm Sho Suzuki from Kao Corporation. I'd be glad to have the opportunity of making this presentation. Today, I'll talk about epidermal sensitization assay named Epicensor, which is an in vitro skin sensitization assay based on gene expressions of reconstructed human epidermis. So this slide shows today's topics. We have two speakers at this presentation. At first, I'll talk about these two topics. After that, Dr. Mizumachi will talk about another two topics. So introduction. Allergic contact dermatitis is one of the dermatological diseases caused by skin sensitizers and bring on a significant psychological effect to patients. In addition, once someone sensitized, the patient needs a lifelong protection from the sensitizer, which causes a decreased quality of life. Therefore, skin sensitization is one of the most important toxicological endpoints. Although skin sensitization used to be assessed by animal testing, because of animal welfare and regulatory requirements, development of non-animal test methods has recently been stimulated. The process of skin sensitization has been described as adverse outcome pathway in the report published from OECD. Briefly, the process includes several key events. Key event 1 is binding to biological protein. Key event 2 is keratinocyte response. And key event 3 is dendritic cell activation. To assess the skin sensitization of various chemicals, several kinds of in-chemical and in vitro assay focusing on these key events have been developed. Recently, 7 methods based on key events of skin sensitization have been adopted as OECD test guidelines. However, there are some common limitations at existing alternative methods. One is lipophilic chemicals. In animal testing, lipophilic vehicles can be used for dissolving test chemicals, and the test chemical solutions can be directly applied on skin. On the other hand, existing alternative methods use medium or aqueous buffer. Therefore, lipophilic test chemicals may cause a phase separation or precipitation producing, which may lead to insufficient exposure and result in false negative. And another limitation at existing alternative method is preplohaptens. They need to undergo autooxidation or metabolic conversion in order to become sensitizing haptens. Because these cell lines have only limited metabolic activity, and both DPRA and ADRA have no metabolic capacity. So existing alternative methods may have less predictivity for preplohaptens. To overcome these limitations, we focused on reconstructed human epidermis or RHE model. There are many advantages to using RHE model, such as metabolic similarity to human skin, and lipophilic chemicals can be directly applied on RHE model like animal test. In addition, several commercial RHE models such as EpiSkin, EpiDerm, Skin Ethic, and the Labocyte EpiModel have adapted to OECD test guideline. From these reasons, RHE is expected as a sufficient tool with wide applicability domain like animal test, and we developed RHE model-based assay called EpiSensor to mainly evaluate about key event 2. At the key event 2 of skin sensitization, keratinocytes are stimulated to activate antioxidant response genes and to release danger signals, for example, pro-inflammatory cytokines or ATP. This figure shows the selection flow of marker genes. First, sensitizers or non-sensitizers were applied on RHEs, and the microarray was performed to find the candidates of marker genes. Second, real-time PCR was performed to select the effective candidates. At this time, 3 selection criteria were set. That genes that showed significantly marked induction by sensitizers, but not by non-sensitizers. In addition, the relevance to keratinocyte response at skin sensitization and the reproducibility in both microarray and real-time PCR were also considered. At last, ECBAM reference 16 chemicals were tested, and 4 marker genes called GCLM, DNA JB4, and IL8, and ATF3 were selected based on performance for discriminating sensitizers from non-sensitizers. GCLM serves as a key determinant of biosynthesis synthesis for GSH, which can regulate redox signaling and antioxidant response. In addition, induction of GCLM gene regulated by both NRF2, ARE, and AP1 pathways like these figures. Next, DNA-JB4 is known to suppress protein misfolding by oxidative stress as chaperone. And induction of DNA-JB4 gene regulated by both NRF2, ARE, and HS1, HSE pathways. Therefore, these genes relate to cytoprotective responses of keratinocyte. Next, IL-8 serve as a potent chemotactic peptide for neutrophils, which are critically involved in both sensitization and elicitation phase of contact hypersensitivity. Furthermore, induction of IL-8 gene regulated by ATP, P2X7, and P38 MAP kinase pathways. ATF3 serve as a hub of cellular adaptive response network by negatively modulating inflammatory cytokine and chemokines. Therefore, the induction of ATF3 gene regulated by ATP and NF-kappa B. Therefore, these genes relate to inflammatory of keratinocyte. The relationship between marker genes and key regulatory molecules have been experimentally confirmed. Regulatory molecules such as NRF2 and P2X7 were knocked down by siRNA and antagonist, respectively. As a result, DNA-JB4 gene expression induced by DNCB was decreased by siRNA. And a similar result was obtained in GCLM. In addition, ATF3 gene expression induced by DNCB was decreased by P2X7 antagonist. And a similar result was obtained in IL-8. In summary, expressions of GCLM and donor JB4 are partially regulated by NRF2, and the expressions of ATF3 and IL-8 are partially regulated by P2X7. This slide shows the outline of EpiSensor protocol. First of all, solubility check of test chemicals is performed to determine appropriate vehicle and the highest concentration at dose finding study. The second step is dose finding study to determine tested concentration at the main study based on cell viability. Finally, main study is performed to judge the test chemical as positive or negative. Regarding solubility check, an appropriate vehicle is selected from 3 candidates, such as AOO, distilled water, and 50% ethanol. The highest test concentration is 100% and 50% for liquid and solid chemicals, like animal test. In addition, test concentrations are decided based on cell viability at the dose finding study. At main study, a test chemical is dissolved to selected vehicles and 2 whole serial dilutions is performed. After that, 5 microliters of solutions were exposed on the surface of RHE model in triplicate. After 6 hours of exposure, the RHE model collected and the real-time PCR is performed to measure the gene expression. In addition, LDH assay is also performed to measure cell viability. After that, at least 1 marker gene exceeds respective cutoff value, with cell viability is higher or equal to 80%, that chemical is judged as positive. This table shows the predictive performance for 69 lipophilic chemicals. These lipophilic chemicals were selected based on the data of animal test called LLNA, variety of high physicochemical properties and commercial availabilities. The result of EpiSensor is compared to those of existing 3 test methods adapted to OECD test guideline called DPRA, keratinosense, and HCRAT. This number of interpretable chemicals means the number of chemicals which can be judged as positive or negative. In addition, this table shows the predictivity of EpiSensor for 67 hydrophilic chemicals. As you can see, a lot of chemicals cannot be evaluated in existing method, especially in keratinosense and HCRAT. But all tested lipophilic chemicals can be evaluated in EpiSensor. In addition, as you can see, accuracy for lipophilic chemicals is higher than these existing test methods. Therefore, EpiSensor could predict sensitizing potential of lipophilic chemicals and hydrophilic chemicals with high reliability. Next, this slide shows the predictivity to 37 preprohaptens defined by published reports. As you can see, almost preprohaptens could be correctly detected. And EpiSensor showed better predictive performance than existing test methods. The next slide is overall predictive performance based on 136 chemicals, including 69 lipophilic chemicals and 37 preprohaptens. It is clearly shown that EpiSensor had high predictive performance. Sensitivity is 88%, specificity is 66%, and accuracy is 83%. And also shown broad applicability domain to various chemicals, including lipophilic chemicals and preprohaptens. Secondly, I'd like to talk about predictivity at human data. This table shows the predictive performance of LLNA, which is animal test, EpiSensor, and existing methods to 80 chemicals. It is confirmed that EpiSensor showed predictive performance similar to LLNA. On the other hand, specificity of EpiSensor is also similar to LLNA, but lower than other test methods. This slide shows the false predicted chemicals at human data. As you can see, almost chemicals are reported as human potency class 5. At Dr. Basqueta's publication, it means very weak sensitizer. Moreover, almost chemicals were judged as positive in LLNA. From these reasons, majority of false predicted chemicals may have weak sensitization potential. Next, internal validation study of EpiSensor was performed under JACBAN. As you can see, a lot of experts were participated as validation management team. In addition, 3 Japanese laboratories were participated in this study. As a result of a validation study, within laboratory reproducibilities based on 15 chemicals were met the target criteria at all laboratories. And between laboratory reproducibility was also met the criteria. From these results, the validation study was completed, and it was confirmed that EpiSensor was a robust and reliable method. The next section will be presented by Hideyuki Mizumachi. So now I'd like to hand over to him. Are you ready?
Okay. Please wait a minute to show my screen.
Dr. Suzuki, you have to stop sharing. Then Dr. Mizumachi will be able to present.
Next, Hideyuki Mizumachi will want to share my presentation. Can you hear me? And
yeah, Dr.
Can you
Suzuki
see my presentation?
Sharing. Dr. Suzuki, please stop sharing.
This slide is shared by Mizumachi.
Okay.
So.
Okay. Okay, so hello everyone. I'm Hideyuki Mizumachi from Kao Corporation. First of all, it is a great pleasure to have the opportunity of making the presentation this meeting. So next I'll talk about the EpiSensor-based defined approach as the next topics. As you may know, as Dr. Hajime Kojima said, OECD guideline number 497 is one of the big topics of this year. This is the first OECD guideline that can be used to address countries' requirements for hazard and potency identification. This guideline contains 3 defined approaches, which are based on the result from multiple information sources to provide information that is equivalent to animal studies. One is 2 out of 3 defined approach for hazard identification based on the result of DPLA, keratinosense, and HCRAT. The others are integrated testing strategy, or ITS, for hazard and potency identification. ITS version 1 is based on the DPLA, HCRAT, and DEREC. This is INSECO tool. And ITS version 2 is based on DPLA, HCRAT, and OECD QSAT toolbox as a INSECO tool. Therefore, the replacement of animal tests for skin sensitization may be more advanced than that of other toxicological endpoints. However, the common limitations of in vitro methods, such as lipophilic chemicals and preprolactants, may still remain in defined approaches, because the limitation of those are based on the limitation of individual information sources. Therefore, we thought that the development of EpiSensor-based defined approach may be a promising approach to solve these issues. At this section, I'll talk about the potency prediction using EpiSensor at first. And after that, I will explain about EpiSensor-based defined approach for skin sensitization hazard and potency identification. Regarding potency prediction, we focused on EpiSensor minimum EC values, that is estimated lowest concentration resulting in positive in EpiSensor. This figure shows the relationship between EpiSensor minimum EC values, that is vertical axis, and LLNA EC3 values, horizontal axis, for 89 chemicals. It's positive in both LLNA and EpiSensor. Here, LLNA EC3 value is widely used as the index of skin sensitization potency, and 2% is the approved criteria to distinguish GHS category 1A, that is strong sensitizer, from GHS category 1B, that is weak sensitizers. We tried the categorization of sensitizers like LLNA and investigated the criteria which can minimize the number of under-predicted and over-predicted chemicals. As a result, we found out that the 0.1% of epicenter minimum EC value is a promising criteria to predict potency. From this result, the potency classification was performed based on the concentration showing positive marker gene expression of epicenter. When the lowest test concentration showing positive gene expression is higher or less than or equal to 0.1%, the chemical is judged as strong epicenter. And if more than 0.1%, the chemical judged as weak. And of course, if all marker genes don't exceed the cutoff values, the chemical is judged as non-sensitizer. These figures show the example result of epicenter. The vertical axis shows fold induction, and horizontal axis shows test concentration. And this green line shows respective cutoff value for 4 marker genes. From this figure, the lowest test concentration showing positive gene expression is less than 0.1% at ATF3. So this chemical is judged as strong in epicenter. This table shows the result of potency classification based on 136 chemicals, including a lot of lipophilic chemicals and prepolhaptens. As you can see, the potency accuracy versus LLNA was 71.3%. So from this result, we think epicenter has favorable performance for not only hazard prediction, but also potency prediction. Next, I'll talk about the development of epicenter-based defined approaches. Here, several key points about mechanistic coverage of defined approaches are shown in supporting document of guideline number 497. Defined approaches should be mechanistically relevant to the existing skin sensitization AOP framework, and at least one key event should be covered at that defined approach. Epicenter is based on key event 2, keratinocyte response. So the method based on other key events such as DPRA for key event 1 or HGRAD for key event 3 might be a candidate to combine with epicenter. However, as Mr. Suzuki presented, these methods have the limitations for lipophilic chemicals. Therefore, we thought a method that is unaffected by chemical lipophilicity is more appropriate. So we focused on the in silico tools. Today, I'll explain about the combination with Times skin sensitization tool that is commercial tool from Oasis. The Times is a QSAR model with skin metabolism simulator, and the protein binding is predicted based on the chemical structure. In addition, the Times can predict skin sensitization potency in strong or weak. So this novel defined approach based on combination of key event 2, epicenter, and in silico tool is called epicenter ITS. Epicenter ITS is a simple score-based system based on the potency prediction of epicenter and in silico tool. This slide shows the prediction model and result. First, the result of epicenter and times are converted into scores. As you can see, regarding epicenter, resulting in strong, weak, and non-sensitizers are converted to scores 3, 1, and 0 respectively. Likewise, regarding times, resulting in strong, weak, and non-sensitizer is converted to 2, 1, and 0 respectively. Here, the score of strong result in epicenter is higher than that of times. This is because epicenter was more effective to detect strong sensitizers than Times insect tool based on the result of over 100 chemicals. The second, the total score is calculated and overall prediction is performed based on this table. This prediction model is developed to maximize the hazard and potency accuracies. This table shows the result of Epicenter ITS. Regarding hazard prediction, the sensitivity, specificity, and accuracy were 83%, 91%, and 85% respectively. In addition, the potency accuracy was 74.1%. As a result, Epicenter ITS shows favorable performance for both hazard and potency prediction. Next, the predictive performance of Epicenter ITS was compared with existing defined approaches, scoring 2 out of 3 or ITS version 1, using 136 chemicals, including a lot of lipophilic chemicals and preprohaptens. Here, regarding 2 out of 3, overall judgment is based on the 2 concordant results of DPLA, keratinosense, and HCLAT. In addition, ITS version 1 is a simple score-based system like Epicenter ITS, and that is depending on DEREC, tool, and DPLA and HCLAT. As you can see, there is a lot of chemicals which are not interpretable in 2 out of 3 or ITS. On the other hand, almost chemicals were applicable to Epicenter ITS. In addition, the predictive performance for hazard and potency identification was comparable to existing defined approaches. Next, we tried to use free in silico tool called OECD QSAT toolbox instead of Times. This table shows the result of Epicenter ITS version 2 based on Epicenter and OECD QSAT toolbox. And comparison with comparison with the existing defined approaches. Here, ITS version 2 is based on QSAT toolbox, DPLA, and HCLAT. As you can see, almost chemicals were applicable to Epicenter ITS version 2. In addition, we confirmed that the predicted performance for hazard and potency identification of Epicenter ITS version 2 was also comparable to existing defined approaches. This is a summary of our presentation. Epicenter is in vitro skin sensitization method based on gene expression of 4 marker genes, which are related to keratinocyte response of AOP. In addition, Epicenter can predict skin sensitization potential of various chemicals, including lipophilic ones and preprohaptens, with high accuracy. Moreover, the validation study of Epicenter was completed with sufficient results. And at last, Epicenter ITS have broader applicability domain than existing defined approaches and sufficient predictive capacity. At last, this work was supported by a lot of collaborators, especially at validation study. I'd like to appreciate them. Thank you very much for your kind attention.
Yeah, thank you very much for the presentation, Mizuma-san and Suzuki-san. Okay. So, do you have any comment and question to this presentation? So, I would like to first, my question is I'd like to clarify that to applicability domain. So
Yes.
To this assay that to available to detect the preprohapten and lipophilic test chemical. The
Yes.
other out of the other chemical is out of applicability domains. To please just tell me that to audience.
Okay, thank you very much for your for your question. So we think there is some technical limitations for some chemicals, such as this method have three candidates of vehicles such as AOO, 50% ethanol, and distilled water. But if the test chemical is not solved or do not form a stable dispersion at very low concentration at 0.012% in these three vehicles, the chemical is not applicable in this method, we think. And this this method is based on the gene expression assay of RHE model. So we have to elute the messenger RNA from RHE model. Therefore, some test chemicals that affect RNA itself, such as causing RNA degradation, or the RNA isolation mechanism itself, that chemical might not be suitable for testing, I think.
Thank you so much.
Yeah.
Okay. Do you have any comment on that question?
Yeah, I just have one.
Oh, please. Yeah.
Hello, this is Mani, Dr. Wangala. Just one small, it's not a question, it's just I'm trying to understand myself. What does over-prediction and under-prediction mean? Because, I mean, I think these are in vitro assays. You predict or don't predict to some extent, it's fine. But what is over-prediction? What is under-prediction? In humans, we're all different. I mean, every human is different. Every ethnic group is different. So you may over-predict for one ethnic group, may actually predict correctly to another ethnic group. But I'm not understanding the terminology. Is that a regulatory terminology or your terminology saying that over-prediction, under-prediction?
Thank you for your question. At this presentation, over-prediction and under-prediction means the 2 versus LLNA means 2 versus LLNA. If the LLNA judged some test chemical at category 1A or a strong sensitizer, but epicenter ITS judged that chemical as category 1B or not classification, the chemical, we think the epicenter ITS under-predicted the chemicals. And of course, LLNA judged some chemical at category 1B or not classified, but epicenter ITS version 2 judged the chemical as category 1A. We think epicenter ITS version 2 is over-predicted the potency of these chemicals. This is answer for you. Is it okay?
Yeah, fine. I'll talk to you later. Okay, thank you.
Okay, thank you. Do
I
you
have a question, if I may.
Please. Yep.
Thank you very much for your presentation. Very interesting.
Yeah.
I have a question about the applicability. Did you test not chemicals, but finished products such as pesticide, medical devices in your essay?
Yes. Thank you for your question. Yes, this method, epicenter or epicenter ITS, is for only chemical, pure chemical, not a product, we think. But there is some study is, we believe some mixture or product can be tested and get some applicable or reliable result will be obtained for such as the medical device or cosmetic product and so on. That is my belief. Yeah. But
Perfect.
basically, this method and efficiency ITS, the defined approach is for pure test chemicals.
Yeah. Okay. But
Okay? Yeah.
thank you very much.
Yeah, Christian, this assay is very so useful to evaluate the medical device in the future.
I'm sure of this. I'm sure, Hajime. We will have an opportunity to discuss. Thanks very much.
Thanks so much. Yeah, thank you for the question. Okay, time is limited, so I would like to close this presentation. Is okay? So, Patty.
Yes, sir. Yes, sir. Thank
Yeah, very good presentation, technical presentation.
Okay.
I must appreciate.
Yeah, Lasudin, thank you very much.
Yeah.
So, on behalf of JSAE, I appreciate.
They need to do proper marketing. It's still such tests, and the company the agent they have hired in India, it's not yet much popular, or they have to do proper marketing for their products, for their services. In fact, even I am not aware, though I have some interest in testing, clinical, preclinical testing. So I think our friend from Japan, they have to insist their Indian partners to more popularize and conduct maybe some workshops, product-related workshops in academic institutions where they can demonstrate their products and also align with industry. So both the presentations were very good and very, I think the technology is very good, and we need to see that if they become much popular in our Indian scenario. So I must congratulate my friend from JTech and hope that it will be much popular and much used in. And Japanese products are always cheap, and we are very happy with their quality control. So good, Dr. Hajime, bringing these guys to our platform.
Thank you, Lasudin.
Thank you, Dr. Rajni.
Yeah.
Thank you very much.
Yeah, we are almost 10 minutes low by time. Yeah. So I'll request now Dr. Hajime Kojima to hand over the dais to our next chairperson, Dr. Date. Dr. Date is there on board, I think. Hello. Sorry, Dr. Wangala. Dr. Wangala, please.
Hi, I'm here. Thank you. Thank you for handovering this to me. Actually, you know what? Before I start introducing Dr. Christian Palabosian, Dr. Vijay Paul Singh had a few things to say about the FASAI and Ayush. I would like to give him an opportunity to do this right now because I'm curious about the FASAI and Ayush organizations. Dr. Paul, would you like to say a few things about the FASAI and Ayush? How do we approach these two different?
Yes, sir. I hope you can hear me.
Yeah,
you're audible.
Yes.
Yeah, can you? Yeah, okay, sir. Sir, just I want to highlight there are three things into this. If there is a approval for the food, then the FSSAI is the agency. If there are approval for phytopharmaceutical, which needs 4 ingredients, then it is CDSCO. If it is Ayurvedic, then it is the state government which is dealing with this registration of the medicine. But there is a lot of overlap in this one. Then what is, there is a task force which is being formed. I have also shared the PPT that what is nutraceutical, what is food for special purpose, dietary usage, food for special medical purpose. FSSAI only approve the nutraceuticals which are already there in Schedule 8. If they are not in the Schedule 8, then there is novel drug which have a separate approval. But just I want to say, sir, there are many overlap in between Ayurvedic, nutraceutical, as well as the drugs which are phytopharmaceutical, and latest was the coronil. So I can just say there is a committee which is being formed. If it should not have more than one RDA. If there is more than one RDA, then it goes to the special food for special medical purpose. And also, if I have some indication, which is a drug, then it goes to either CDSCO or Ayurvedic state pharmaceutical, this one approval authority. So there is a lot of overlap, but still, I can cite the example like Revital is approved as this supplement. So this is a cognizance taken by the government, and there is a committee which is working on this RDA, how much to be allowed based on ICMR guideline, and what is Ayurvedic, what is nutraceutical, what will go where. And just I wanted to highlight this part. Thank you, sir.
Yeah, just one quick question. About a few days ago, I got a note from somebody saying that N-acetylcysteine, which is also considered a nutraceutical product, recently Amazon pulled out all those products. They're not selling them anymore. The reason for that is because FDA questioned, because it was an approved drug several years ago. Now it cannot be considered as a nutraceutical. So there's some overlap also.
Yeah.
Definition. Yeah.
If I may add, this is Amir here, Dr. Mangala. So, and thanks, Dr. Vijaypal Singh, for that clarification. So, regarding the nutraceuticals, we have this one mission which is run by CSIR on nutraceutical mission. So, there we have representatives of FSSAI as well. There is this Gazette notification wherein FSSAI has, Dr. Vijaypal Singh is aware of that. I mean, there are some 300 ingredients that have been enlisted over there. So, those ingredients, if given within those recommended dosages that are, it's almost like, say, if we talk about curcumin, which we anyway take as part of turmeric. So if there is turmeric within those permissible limits, which we anyway take, so those are the ingredients that go into the nutraceutical mode, because it's considered that we are anyway taking those, and there are known, I mean, kind of human data that it does not have an adverse effect. So those 300 ingredients, as per FSSAI and Government of India's Gazette notification, so those ones actually go into the nutraceutical domain. And even if, I mean, there is a very stringent kind of domain within that also, if the solvent is changed. For example, it's said that if they are extracted by aqueous and ethanolic extract method, as long as, even if the solvent changes, for example, if you go to hexane and chloroform based extraction, then again, it will be going into the different domain. So yes, so that's preliminarily.
I think Dr. Kishan is waiting for us to get muted. Okay.
Okay.
Thank you. Sorry to disturb.
Yeah.